Hello, everybody. I hope you guys enjoyed learning a little bit about cell membranes and how we can specifically select only certain things to come into and out of our cells from the assorted videos that I posted for you guys. In general, hearing about any scientific concept from multiple perspectives will definitely help you to understand. And so this particular video will include me introducing you to the lecture activity that is associated with this particular lesson today. So you learned, of course, about diffusion, right? So a few different types of diffusion, and you learned about active transport, including both the use of pumps as well as the use of endo and exocytosis. Okay. The lecture activity today is going to involve you accessing a portion of mastering AMP and essentially parsing out some of the factors that affect diffusion and active transport. There will be 3 activities. In the first activity, you're going to start learning about what actually affects the rate of diffusion. And so in our very next lesson together, we're going to learn about how neurons work, and you'll see that it is critically important for the function of the neuron, and therefore the function of the entire body, to actually change the permeability of a cell to different types of ions. That is, change how much diffusion can actually happen depending on what is going on in the body and the cell at the time. So 2 factors that I can tell you right now that determine the rate of diffusion, that is, that can increase or decrease the diffusion across a membrane into or out of the cell. First of all is temperature, right? So essentially, the more you increase the temperature, the more Brownian movement, as the one video talked about, the more Brownian movement is going to occur. Therefore, these molecules or ions are moving more quickly, and therefore they are going to diffuse faster. Okay, so essentially, if you spray the bottle of perfume in a warm room, on the opposite side, you'll be able to smell that perfume faster than if you were in a cold room. Okay. Something else that was brought up by the videos today is that essentially the size of the passenger is actually really important. So not only is it going to determine what transport mechanisms are employed, but also the bigger and clunkier the substance you're trying to move, the longer it actually takes for that substance to diffuse across the membrane. And so there are actually 2 more factors that affect the rate of diffusion across the cell membrane that you are going to be kind of playing with experimentally in your exercise today. In activity 3, you're going to learn about osmotic pressure, right? And so again, you are going to kind of apply different conditions, different levels of variables, and see when osmotic pressure is increased or decreased. Okay, and finally, in activity 5, you're going to be learning about active transport, specifically the sodium and potassium pumps. These are certainly not the only pumps that are present and important in your cells, but they are certainly the ones that we talk about the most. And so you're going to clarify in your mind what factors affect these pumps, as well as what gradients they're going to produce. Okay, and so let me walk you through what you are to do today. Okay, so on Blackboard, in addition to the links that you guys have, you have a Word document with both questions and instructions for how to proceed through this, this assignment. Okay, so essentially you're doing 3 different activities or portions of 3 different activities. Those are highlighted in yellow for you. Okay, and the questions you are to answer are highlighted in red like this. Okay, so just trying to make it as clear as physically possible. And so you will access Mastering AMP. Right, normally you see your assignments, but today I want to point out that you have a study area. Okay, and so this study area has a ton of different things available to you. And just before we get into today's exercise, let me show you that you can study by chapter, right? So there are little quizzes, there are videos, right? Here's one on membrane transport, how relevant, to help you prepare for upcoming assessments, okay? Also, if you are trying to study for the histology lab quiz that's coming up at the end of this week, Practice Anatomy Lab is a program that I think is excellent. You can look at anatomical models, which will become a little bit more important later. And what is relevant to us now is histology, right? So you can go in here, looking at epithelial tissues. Here is some histology in addition to the lovely pictures that you guys provided on Google Slides. But here you can, of course, zoom in, right? And there are labels associated with these, right? So this is a kidney, right? And you can see different types of epithelial tissue here. Okay, different types of slides, again, lots of labels so that you can actually tease apart what is connective tissue versus what is the epithelial tissue that you're actually trying to look at. And then of course here is the blood, so you can see actual red blood cells as well. And so if you want to test yourself, right, there are also little quizzes, right? What is this particular section that is highlighted in this particular slide? And right, it'll give you the answers if you would like, and so on and so forth. So definitely a really helpful tool for you guys, right? However, for now, your goals for the day are to, of course, finish this activity in PhysioX. Right, so again, there are lots of different experiments that you can watch that are really interesting, but today we want to actually complete exercises. Okay, so you are looking at exercise 1, so this first set up here. Later in the week, we will work on these down here. But for today, you're starting off with activity number 2. Now, in this activity, there is a lot of kind of background information that will really help you to understand what exactly you are seeing in this exercise. You are more than welcome to read it. You're more than welcome to complete the pre-lab questions, etc. However, all of this stuff is not mandatory. You don't have to hand in any of that kind of information. Instead, you can simply skip right to the experiment if you want. Okay. And so here is the experiment. Note that there are instructions up here. So this is instruction number 1. And so if we look over here in your lab handout, what you can see is that you are to follow instructions number 1 through 7. And so just to give you a little bit of background here, these 2 beakers represent kind of inside versus outside of a cell. And so we are inserting a membrane according to the instructions. And so the first step here is to set this membrane to 500 glucose carriers. Right, you're going to build it, and ultimately you are going to slide it over in between these 2 beakers. Right, when you're done, you click next. Right, so dragging the membrane, and again, this will be very thorough about telling you exactly what to do. Right, so now these 2 environments are separated by a membrane, and this particular membrane has 500 glucose carriers. So these are going to allow for the passage of glucose over this membrane, right? Of course, with the concentration gradient, this is a passive process. Also, this is facilitated diffusion, implying that, again, this is passive transport, so it doesn't require any energy. Substances flow with their concentration gradient, and also it is facilitated, right? So glucose can't just cross over the membrane at will, right? In our plasma membranes, it would be stopped by those hydrophobic lipid tails, right? So we need to facilitate this, in this case, with 500 glucose carriers in this membrane. Okay, so now we need to start adding stuff to these beakers. So here we're going to add 200 millimoles to the left beaker in particular. So for glucose, we add 200 millimole, or 2 millimoles, we dispense that fluid. All right, going on to the next thing, we're going to add deionized water. So essentially this doesn't have any ions inside of it, or any, glucose inside of it. It's just plain old water. Okay, and so then we are going to start this trial. Look how the timer has minutes, right? This is essentially fast forwarding through an hour, right, in only a few seconds. And so what we see here is that glucose reached an equilibrium at 43 minutes. In other words, if you watched over here, we start out with 2 glucose on this side and 0 over here. And so glucose flowed down its concentration gradient to this beaker over here, again, through this particular membrane. And so when equilibrium is reached, we have equal amounts of glucose, 1 millimole, on each side. And so this time to reach equilibrium is one of the dependent variables that we're actually going to be measuring today. And so we can take this 43 minutes over to our chart over here. And so we had 2 millimoles of glucose, 500 glucose carriers. And so the time it took to reach equilibrium, we can just type in that it was 43 minutes. Okay, and so you will be doing exactly this when you proceed with this experiment. Okay, if you read these instructions over here, you will see that you are to flush the beakers out. All right, so essentially starting over again, you can keep this 500 millimole membrane, and essentially you're going to be completing that step over and over again. So now the next in line here is 5 millimoles, so we'll add 5 over here. We will add DI water on this side and ultimately start all over again. Okay, and so you're going to complete this process over and over again until you can fill in this chart. And of course, in my haste, I forgot to mention one step. Okay, so glucose equilibrium is reached in 52 minutes. All right, so here we reached equilibrium in 52 minutes. And what I forgot to mention, I apologize, is that you should record the data in between. And so the glucose rate of diffusion is 0.0016. Okay, so 0.0016 millimoles per second. Okay, and so again, you're going to just keep on filling in this chart here. Okay, ultimately you're going to change the amount of glucose carriers in the membrane to greater and less than this 500. Okay, note when you look over here, when you use this 300 millimole membrane, you should be certain to increase the timer, right? Otherwise you will not reach equilibrium and you will not be able to collect the data that you need for this particular experiment. Okay, so when you have completed this particular chart, don't X out of this yet. You still need to use that one more time. Okay, next you're going to graph this information. Okay, and so what you can do is simply copy the graph into whatever program you like to draw with, or even if you can draw within the Word document somehow, by all means, do whatever you do. But I just want to show you that you know, you can easily, you know, make your scale. All right, so make sure you type in labels for the y-axis, and I'm just kind of making this up as I go along, right? I don't really have the data available, but here you can add in, add in these shapes, right? So if you have, you know, whatever, this is a data point, right? So here's one data point, and here is the next data point, again, looking and graphing this appropriately. And your next, oops, your next data point, right? And ultimately you will draw a line between them to make it a little bit easier to see. And I will say that the data that I have been graphing is for the 300 glucose carrier data, okay? And so ultimately, what I would like to see is a graph that looks kind of like this, right? Make sure that you graph the number of carrier proteins and, oops, actually, sorry, that would be It's not the carrier proteins. In fact, it would be sorry about that. Instead, sorry, this would be 8 millimoles of glucose, right? This set of data would be 5 millimoles of glucose, and this would be 2 millimoles of glucose. Okay, and so what this type of graph will let you do is look at both how the concentration affects the time it takes to reach equilibrium, and it will also help you to analyze how the number of glucose carriers in the membrane is going to help you, or is affecting the time it takes to reach equilibrium. Okay, and so you can do something like this, and then just copy and paste this back into the Word document. Alternatively, if you want to get really fancy, you can actually use Excel or Google Sheets or something to graph this data for you. Whatever you want, it doesn't have to be a work of art. You could literally sketch it out on a paper and insert an image of that. Don't care, as long as you give me a graph that looks something kind of sort of like this. And so that's important because you will be talking about these results, right? So essentially, the goal of this particular activity is to figure out what factors affect the rate of glucose diffusion, or just diffusion in general. And so you are going to talk about the results that you got from this Mastering A&P program in terms of glucose carrier abundance, right? So as the number of glucose carriers increases in the membrane, the rate of glucose diffusion across that membrane is going to increase or decrease. Okay, so that is what you will be talking about here. Okay, next, you are going to go back to Mastering A&P, and this time you're going to try putting glucose on both sides of the membrane. Okay, so if you were following directions 1 through 7 over and over and over again, you would have been putting glucose over here and DI water over here. But now you're going to put 2 millimoles on the left and 5 millimoles on the right. Okay, and so once again, you are going to run the same experiment, right, with the 500 millimole membrane, right, and then put the time to equilibrium here. Okay, once you have done that, you can then restate your conclusions, right? It's okay if the conclusions that you got up here aren't exactly correct, because the goal here is for this particular experiment to clear up this situation. And so here, glucose equilibrium reached at 47 minutes. Once again, you can just type this in. Okay. Okay, so that is activity number 2. Activity number 3 is osmotic pressure, and so in order to access that, you should go back to this PhysioX screen, okay, and go into activity number 3. Again, you can skip all this extra stuff. I'm not going to see that. You are welcome to do that. It will hopefully help you to understand, but for my purposes, for our purposes as a class, go right to the experiment. Right, and again, you're going to be following the directions, right? Here the membranes are made for you. And again, you are adding the stuff they tell you to, and then running the experiment. Now you only have to do numbers 1 through 16. Right, that is, go through these instructions 1 through 16. Sometimes they ask you little questions in there, and these questions, again, are not going to be given to me by the program. However, they might help you to actually understand what you're seeing. And so, as always, you're going to be recording your data. You are going to be looking at pressure, right? We're looking at osmotic pressure here. So essentially, so you're looking at osmotic pressure, and you're going to be answering these questions about what you learned from this exercise. None of this data that you collect down here is something that you need to give to me, but it will be important to actually collect it so that you can talk about it in these questions here. Okay, finally, you are going to be looking at active transport. Okay, again, you don't have to answer any of the questions, do the pre-lab, none of that stuff. Just go right to the experiment. Keep in mind that for this particular experiment, you are looking at what is happening inside the cell versus what is happening in the extracellular environment. You will be recording data and going through all of the instructions up here at the top. You will not have to cite individual numbers that you collect here, but again, it will really help you to have this data in front of you when you start to answer these questions here. Okay, so when you are finished with this exercise, please save the file and submit it to me on Blackboard via that assignments link. Okay. Thank you guys very much. I look forward to talking about this information with you guys on Wednesday in class, where we're actually going to apply this diffusion and active transport and all of these different concepts that you learned today to neurophysiology. So really finally getting into anatomy and physiology at this point, talking about how neurons work to actually control our body. Okay, so thank you guys very much and have a wonderful day.